Imagine a world where the very blueprint of life can be rewritten at the push of a button. We're not talking about science fiction or futuristic fantasies. This is the reality of today's genetic engineering technologies. In the world of AI-driven biology, we're venturing into uncharted territory where machines don't just predict protein structures or design new genetic sequences, they hold the power to disrupt life itself. Join us as we look at some of the most disturbing applications of AI in biology. To kick things off, let's talk about something you might have heard of: Google's AlphaFold. Now, AlphaFold is an incredible AI system developed by DeepMind, a Google-owned company. It's designed to predict protein structures, which, for those not deep into biology, is a really big deal. Proteins are like the little machines in our bodies that do pretty much everything, and knowing their structure helps scientists understand how they work. So, why is this so revolutionary? Well, before AlphaFold, figuring out a protein's structure was a slow, painstaking process that could take years. But now, thanks to AI, scientists can do this in a fraction of the time. It's a game changer for biology and medicine because it means we can develop new drugs and treatments much faster. But here's where things get a bit spooky. With great power comes great responsibility, and the power of AI like AlphaFold brings some pretty disturbing possibilities. Imagine if someone used this technology for not so noble purposes. Instead of creating life-saving medicines, they could design harmful biological agents. This isn't just science fiction. The fear of bioterrorism becomes more real when you have powerful AI tools that can be misused. Let's get specific. AlphaFold can predict the structure of proteins with incredible accuracy. While this can help in developing new treatments for diseases, it also means that someone with malicious intent could use it to design proteins that act as toxins. Such a toxin can be designed to target specific human proteins, disrupting vital biological processes. This could lead to the creation of bioweapons that are more deadly and harder to detect than anything we've seen before. Furthermore, AlphaFold's capabilities could be used to enhance the virulence of existing pathogens. By understanding how proteins fold and interact, a bioterrorist could engineer a virus that is more contagious or more resistant to existing treatments. It's like a virus as contagious as the common cold, but with the lethality of Ebola, a nightmare scenario made more plausible by advanced AI tools. Now, AlphaFold is not the only new AI innovation in biology. There is also evolutionary scale modeling, or ESM3, another remarkable yet potentially dangerous AI in biology. This AI model works based on the principle of proteins as programmable entities, much like computers. Instead of using lines of code, we use amino acids. If we could read and write this code of life, essentially the sequence of amino acids in proteins, we could program biology itself. ESM3 is an AI model designed specifically for this purpose. Think of it as ChatGPT, but for proteins. This model can engineer biology from first principles, similar to how we build machines or write software. ESM3 tokenizes the biological properties of proteins: sequence, structure, and function. The sequence is the protein's text, the order of amino acids. The structure is the 3D shape, which AlphaFold already cracked, and the function is what the protein actually does. ESM3 integrates these aspects, learning to simulate evolution itself. Scaled across billions of proteins and parameters, this model can generate new proteins from scratch. Recently, ESM3 was tested on green fluorescent protein, GFP, those glowing proteins in jellyfish that have become indispensable in biotechnology. ESM3 managed to create a new version of GFP that is only 58% similar to the closest known fluorescent protein. To put this in perspective, it would take 500 million years of natural evolution to achieve this, but ESM3 did it in moments. While this achievement is groundbreaking, it also opens the door to some unsettling possibilities. One of the significant potential risks of ESM3, especially given the fact that it is free and open available under a non-commercial license, is the rise of biohacking and DIY genetics. The democratization of such powerful AI tools for genetic engineering could lead to individuals or small groups, outside traditional scientific institutions, manipulating biological systems without proper oversight. This accessibility can be both a blessing and a curse. The promise of open-source AI tools like ESM3 lies in their ability to accelerate scientific discovery and innovation by making cutting-edge technology accessible to a broader audience. Researchers worldwide can leverage these tools to push the boundaries of biological research, leading to breakthroughs in medicine, agriculture, and environmental science. However, this same accessibility also lowers the barrier for those with less noble intentions or insufficient understanding of the ethical and safety implications. Biohacking, a movement where individuals experiment with biology, often outside formal institutions and regulations, is already gaining traction. Enthusiasts and amateur scientists are increasingly using genetic engineering kits and DIY laboratories to experiment with modifying organisms. With the power of ESM3, these individuals could now design new proteins or even entire biological systems with unprecedented ease and precision. This can lead to ecological disturbances. DIY genetic modifications introduced into the environment could disrupt local ecosystems. A biohacker might release a genetically modified organism intended to solve one problem, only to create another. For instance, a modified plant designed to resist pests might outcompete native species, leading to unforeseen ecological consequences. Another potential misuse involves synthetic biology. ESM3 could be used to create entirely new organisms with unknown properties. While this has exciting applications in fields like medicine and environmental science, it also presents significant dangers. For example, a synthetic organism released into the environment could disrupt ecosystems in unpredictable ways, potentially leading to ecological collapse or the extinction of species. The ability to create life forms with traits not found in nature means we could inadvertently produce organisms with unintended harmful effects. The issue of genetic manipulation is another area of concern. With ESM3, it's possible to design proteins that can interact with human DNA in specific ways. This capability could be exploited to create targeted genetic modifications, potentially leading to gene drives that spread through populations with unforeseen consequences. In a worst-case scenario, this technology could be used to engineer genes that predispose individuals to certain diseases or conditions, effectively weaponizing genetic engineering. But it doesn't stop there. AI in biology isn't just about proteins. We can now alter human genes, all thanks to CRISPR. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is a technology that allows scientists to edit genes with high precision. It's like a pair of molecular scissors that can cut DNA at specific locations. Once the DNA is cut, scientists can either disable a particular gene or insert new genetic material at the cut site. This ability to precisely edit the genetic code has revolutionized genetics and opened up a world of possibilities. Now, how does CRISPR fit into our discussion about AI in biology, like AlphaFold and ESM3? Think of CRISPR as the tool that can edit genes, while AI models like AlphaFold and ESM3 can predict and design new proteins. Together, these technologies form a powerful toolkit for engineering life at the molecular level. For instance, AI can be used to design new proteins or predict how changes in DNA might affect a protein's structure and function. CRISPR can then be employed to make those exact changes in the DNA of living organisms. This synergy between AI and CRISPR could accelerate the development of new treatments for diseases, create genetically modified organisms with beneficial traits, and even help in efforts to understand and combat genetic disorders. CRISPR's precision in gene editing opens up a world of possibilities, but also introduces several significant risks. One major concern is the potential for unintended mutations. Even though CRISPR is designed to target specific areas of DNA, it can sometimes make cuts at unintended sites, causing genetic changes that we didn't plan for. These off-target mutations could lead to unforeseen health issues or genetic disorders. Another troubling aspect of CRISPR is its potential use in creating designer babies. This concept involves modifying human embryos to enhance certain traits like intelligence or physical abilities. While the idea might sound appealing to some, it raises ethical questions about eugenics and social inequality. It could lead to a situation where only those who can afford genetic enhancements have access to them, deepening social divides. The possibility of creating mutant humans is particularly alarming. Editing genes in embryos or germline cells means that any changes are passed down to future generations. This could result in new genetic traits that have unknown long-term consequences, potentially leading to severe health issues or new genetic disorders. Another profound and unsettling risk of combining CRISPR with advanced AI tools in biology is AI-induced evolution. This term refers to the rapid and unpredictable changes in species and natural selection processes driven by AI-guided genetic modifications. AI technologies like ESM3 can analyze vast amounts of genetic data, predict protein structures, and design new genetic sequences with unprecedented accuracy and speed. When paired with CRISPR, which can precisely edit DNA, we have a powerful toolkit capable of fundamentally altering the genetic makeup of organisms. This capability has the potential to accelerate evolutionary processes in a way that is deeply concerning. The most pressing concern is the unpredictability of these changes. Even with advanced AI, predicting the long-term effects of changes to an organism's DNA is challenging. A modification intended to enhance a beneficial trait could inadvertently introduce harmful side effects. These unintended consequences might not become apparent until after the modified organisms have been released into the environment, at which point reversing the changes could be impossible. Lastly, the ethical and moral implications of gene editing are profound. Deciding what constitutes a desirable trait and who gets to make those decisions raises important questions. Additionally, future generations would have to live with genetic changes made today without having a say in the matter, which adds another layer of ethical complexity. But in our next video, we go in depth into the ESM3 model and how revolutionary it is in biology.